afternoon, grade 12s, and welcome to our lesson on plant responses to the environment. I'm Yolandi Gibson, a teacher at Parklands College.
And I'm Matric a teacher at La Rochelle Girls High School in Paarl.
And together we would be helping you guys better understand the concept of plants responding to the environment.
Okay, so let's jump into it.
Let's go.
So on our slideshow, we're going to just do a little quick introduction about plants because ultimately they're organisms too, right? They also have to respond to stimuli in their environment. Now, not necessarily the same stimuli that we experience, but a lot of them do correspond. They also have to respond to temperature and to light. They also have to respond to water. And the sad thing about it is that they can't, like us, just pick up their roots and go to wherever light is best or water is best. So they have to adjust their growth response to the stimulus. So on the slide, basically we're looking at tropic movements when we talk about this. Growth movements of a part of a plant in response to some or other external stimulus. And those stimuli can include many different things, like I've mentioned, temperature, light, gravitation is one of the ones that we look at as well. And a stimulus is any detectable change, either internally or externally, that could bring about a response. And plants actually use growth regulators known as hormones to bring about these growth responses and regulate cell differentiation. And we'll look at some of these. So the growth hormones that we look at are responsible for example, promoting growth, which not only refers to increasing cell division, but also the elongation of cells. So not only are there going to be more cells, but they will actually become longer and bigger. Also the formation of flowers, stems, and leaves, and also the shedding of those leaves. Like abscisic acid is the plant hormone that's more involved with the dormancy in plants. And then the development and ripening of fruit. So the three hormones that you learn about in grade 12 and that we will be looking at today are auxins, gibberellins, and abscisic acid. Each of them are responsible for different functions within a plant. Auxins mostly for tropic movements and then growth. Gibberellins are your kind of stimulating hormone, and then abscisic acid is, like I mentioned, one of the hormones that promotes dormancy in many different facets of the plant's life. Okay.
Right, so our first hormone that we're going to be discussing is auxins, and auxins will be responsible for elongating cells and ensuring that there will be lengthening in the stems and in the roots. But most important of all, their job is to ensure phototropism, apical dominance as well. So we're going to have a look at that in a short while. So let's revise as to where we will find auxins. Auxins will be found in the growing tips of plants and roots. In this image here, we've got our apical bud, which is right at the top of a plant, and this grayish section here at the bottom, that would represent the root. So the two image on the side, that will be the right tip of a tree, and this will be right at the bottom of a root where the auxin, the site of auxin production would be. Right. So when it comes to the functions, as I've already mentioned, we have our apical dominance, our phototropism, and geotropism being the three main units that we will be discussing during our lesson today. But it's really important that you need to understand that when it comes to learning this section, that you should be learning those three different functions as individual silos. The questions that you might experience in the exam would not combine, for instance, apical dominance and phototropism together. These will be three separate strands that you should learn and always treat as individual strands. So the one that we will be looking at first off is called apical dominance. So let's have a look at our information. Right, so first off, we've already established that our auxins are produced right at the tips in our apical buds, and there will be a very high concentration of this hormone. So here we've got our apical bud being represented here, and with the arrow pointing towards it, and then that will be where the auxins will eventually diffuse down the stem. So diffusion will allow for the movement from an area where there's a high concentration, and the further we move down the stem, the lower the concentration will be. Now, apical dominance is all about ensuring that we have a pyramidal shape or triangular shape within our plants. And the idea behind it is that when a plant grows, we want as much surface area of the leaves being exposed to sunlight. This ensures that there are no competition for sunlight resource for photosynthesis, and all the leaves will get an equal amount of sunlight, which is essential for the autotroph. Now, if we had to, for instance, remove that apical bud, in the example here on the right-hand side, we will see that the plant will no longer apply apical dominance, which is all about inhibiting these lateral branches on the side. So removing that inhibition, removing that high concentration of auxins, will result in these lateral side branches developing. And we no longer have a triangular appearance, but we start to get a more bushier plant. So obviously, like we've said, that will ensure to prevent our whole competition for sunlight. This is the ideal situation that we want the plant to look as a triangular pyramid from a distance. But there are some commercial applications to removing the apical bud in a plant. So this is an example on a farm where the farmer is either farming with grapes or apples or whichever fruit that he is busy with. And if you remove that apical bud, you can make the plant grow in a more of a hedge shape. So that will make it quite easy for the workers to go in and pick the fruit off the tree. So there is a time and a place where we would want to remove that apical bud, as is example there. Right, so the process of apical dominance, we're going to run through this step by step. So the red in this image here at the top, that represents the site of auxin production. And then as we've already established, the auxins will allow for the cell elongation to take place. It will be stimulated for cell elongation and cell differentiation, which means that the cells will develop into a particular cell type. So when we have a high concentration of auxins, it will inhibit these lateral branches from developing. So as I've already stated before, as we move away from the apical bud, the concentration of auxins will start to reduce, and the lower lateral branches will no longer have that high concentration of auxins. So that means that they will actually be allowed to grow. Okay, at the same time that we have our auxins, they will also allow for cell elongation to take place, which means that this plant will continue to grow upright and grow taller in length. And as I've mentioned earlier, because there's a low concentration in this area here at the bottom, that auxin will no longer inhibit these lateral branches, and they will be able to grow sideways. So as a result of the top ones not growing, bottom ones are allowed to grow, we will eventually have that triangular shape within our plant. Okay, it's important just to take note that auxins, besides allowing for apical dominance, also allows for cell elongation to take place. Right, so if we have a scenario where the apical bud has been removed, so here we've got our red representing the apical bud yet again. So this is now an example where the farmer wants to have more of a hedge-shaped tree. We remove the apical bud. Now, as this is the source of the auxin concentration, and now we have removed that high concentration of auxin and that supply of auxin to the plant, what we will find is that our lateral branches are no longer inhibited. The consequence of that is that they will start to elongate, grow, and all the branches, irrespective, will allow for elongation to occur. So eventually what we will find is that it will go from a young plant that looks like this to a hedge-shaped plant, as is in the diagram represented.
Right, so the second function of auxins is to allow for tropic movements. Now, you're going to learn about two types of tropism. Phototropism, which photo meaning light, is the movement of a part of a plant in response to a light stimulus. We all have seen that plants tend to orientate their leaves towards the light, which is obvious because that's the part of the plant which is going to do the most photosynthesis, and therefore it needs the best light exposure. Geotropism refers to a plant's response to gravity, where stems tend to bend away from gravity because, again, a stem wants to be exposed to the air and light. It needs to have photosynthetic parts exposed to light, whereas roots tend to grow towards gravity because that's towards the soil where the water and the nutrients are. So that means that different parts of a plant can respond to a stimulus either positively or they can respond to it negatively. Now, phototropism is the first one we're going to dissect a little bit. So we know now that it is the growth movement of a part of a plant in response to a light stimulus. And usually this is unilateral light, meaning, as in the diagram, it's coming from one direction, and now the plant has enough time to grow in that particular direction. Now, as you can see from the diagram here, if this is some other germinating plant which has a root component and a stem component, the stem part will actually grow towards the light, whereas the root will grow away from the light. So we say that stems are positively phototropic and roots are negatively phototropic. Okay. Now, an important thing to remember when we talk about auxins and phototropism is the effect that the concentration of the auxin has in the root and the stem. Yolandi has just mentioned that auxins are produced in the apical meristem of your stem, and it has a very high concentration there. And then it diffuses down the length of the plant. So it means that the concentration is very high at the top, and it becomes less and less as it moves down. This implies that tissues close to the apical meristem are not very sensitive to auxins. They need a high concentration of that auxin to stimulate growth, whereas areas of the plant farther away, like here in the roots, are so sensitive to auxins that even just a little bit of it will stimulate growth. And in fact, very high concentrations of it will actually inhibit growth. So it's important to remember when you're looking at a root, a high concentration of auxin is actually going to inhibit the growth of the root. Lower concentrations will stimulate it. And in a stem, the opposite is true. High concentrations of auxins in the stem would stimulate the growth, but lower concentrations would inhibit it. Now, with that in mind, let's try to understand what happens during phototropism. So here we have an enlargement of the root that is busy bending. And we want to understand, but why is the side there actually undergoing more growth than the side here? Whereas in the stem or the shoot, we see that the stem is also bending upwards, but we see more growth occurring on the lower side than the upper side. And this has to do with what the auxin is doing in response to the light. So auxins, being light-sensitive chemicals, will shy away from light. They concentrate on the dark side, okay, always. When the stem is exposed, when the stem or the root is exposed to light, the auxins will move to the dark side, and there the concentration is high. Then you must remember, so a high concentration on the dark side in a root is actually going to inhibit the growth, whereas a high concentration on the dark side in a stem is going to stimulate the growth. And that's what causes the curvature. So if we go to our slide, we'll see what happens to the auxin concentration. So our light is coming from the top, and if you look at the root here, you'll see that the auxin is going to move away from the light and concentrate on the dark side. And similarly, in the shoot, we see that the auxin moves away from the light stimulus and concentrates on the dark side. Okay, so auxins are like Star Wars, they like the dark side. So what happens in the root then, right? If you had to explain this in an exam, you would start by saying that if we expose the root to unilateral light, the auxins will move away from the lighted side and they accumulate on the shaded side, or the light side and the dark side. And it's very important that you make that distinction in your answers, that you constantly compare light and dark. Where's the concentration high? Where's the concentration low? Where will more growth occur? Where will less growth occur? Don't assume that the examiner can read into your answer if you said it concentrates on the dark side and now more growth occurs, that you're implying less is happening on the other side. Okay. So if we go back to our answer then, you would start the next sentence by saying, so now growth or cell elongation or cell division is not happening as fast on the shaded side. It's actually inhibited on the shaded side, but it's happening much faster on the side that is on the side that is stimulated by the light, because that side is not having enough auxins. And the consequence of this is that the root actually bends downwards, away from the light. Because you can see here we have more cell division, more cell elongation. So this side's actually becoming longer in comparison to this one, which causes the bend. If we look at the shoot then, you apply the same principle, but the inverse is correct. So if you expose the shoot to unilateral light, or the stem, again, the auxins will move away from the light side and accumulate on the shaded side. But the interpretation of this now is different. If they accumulate on the shaded side in a stem, that stimulates growth. That means the shaded side is now going to grow faster. But the lighted side, which has a lower concentration of auxins, will not grow as fast. So now what we have is more cell elongation and division on this shaded side, and this causes the stem to bend upwards or towards the light in this particular picture. Now, you can definitely expect to have an experimental question with regards to this. They could definitely ask you an explanation of it. So, for example, there was a long question last year in 2019 where you had to actually discuss geotropism and phototropism. But they could also typically give you different experiments, and you would have to apply the principles of these to those experiments. So let's look at a few classical examples of phototropic experiments. So firstly, this would be your typical setup, right? This would be the control setup where we have a germinating coleoptile or a shoot tip or something like that that's being exposed to unilateral light. We haven't manipulated it in any way, and the normal situation will occur. Auxins will respond to this unilateral light, move to the shaded side, concentrating there, causing more growth and cell elongation to occur on the shaded side compared to the lighted side, and the stem tip or the shoot tip will bend towards the light. But now what happens if I remove the apical bud? Like Yolandi said, don't start to think about apical dominance in this case, because we have a light stimulus, so you learn this in little boxes. If you see a light stimulus, we're concerned with the tropic effect of auxins. So if you remove this apical bud, you cut it away, it means that no auxins will be present, right? You've removed the source of the auxins. So now no auxins will spread down this little stem. No auxins are there to respond to the light or even cause elongation of cells. So nothing will take place. No bending, no growing whatsoever. All right? Next up, what happens if we covered the stem tip with something that is not transparent to light? So even though we're applying the light stimulus here, it doesn't reach the auxins in the apical tip. So in that instance, it would be similar to actually not having the apical bud, except that now the auxins aren't responding to light, but they're doing the other things that they should be doing. They're still diffusing down the stem. They're still causing growth and cell elongation. So the auxins stay equally distributed throughout this apical bud because they can't respond to the light that's not reaching them. And that now no growth response to the light is observed. The stem won't curve, it won't move towards the light. However, it will still continue to grow straight. Because even though the light stimulus can now not affect the direction of growth, the auxins are there, and they're still going to stimulate cell elongation and growth. So that's different to removing the apical bud. If I remove the apical bud, I remove the effect of auxins entirely. It's not going to respond to the light, it's not going to cause cell elongation. But if I just cover that apical bud, the light stimulus is not observed, but the growth still occurs.
So with the auxins inside the apical bud, how would they be distributed throughout?
So usually what happens if we have light from all sides, the normal thing that auxins do is they just diffuse straight down in a gradient, just straight down. If you apply the light stimulus, or what Yolandi will explain with regards to gravity, you affect where the auxins start to concentrate. So usually it's just a downwards gradient, a diffusion gradient, high to low concentration.
So an even distribution
Yes.
of the auxins. Okay, and I just want to point out on this particular question as well. It appears in some of the past papers, and they either tell you that it's covered with a foil, or they can tell you that it's covered with a black plastic like a little lid over it. But also take note, sometimes they ask this question and they say that it's a see-through plastic cover as well.
Yeah.
So just be careful of this question. Because,
Transparent compared to non-transparent.
Yes, so don't learn that the plastic covering is like impermeable to light. You have to read what the question is telling you and where it's kind of like guiding you towards with this particular question, application question. Right, and obviously they could come up with all sorts of other experiments as well. So let's have a look at a few other examples of this. Right, so in the image that we have here alongside, we've got a plastic barrier. Sometimes they refer to it as a little micro barrier. And as we know, plastic is pretty much impermeable. But they have placed it on the light side of the plant. Now, we know that this is now the apical bud section and the auxins will be produced. So light is now stimulating these auxins to diffuse towards the dark side of the plant. So that barrier that we have here on the side is actually not affecting the auxins whatsoever. So as we said, these little Star Wars troopers, they're now going to go to the dark side, accumulate here, and as a result of it, they bring about faster cell elongation as opposed to the light side here. And quite often, the auxins are actually destroyed on the light side as well. So that might also appear in some of your past paper memos as a possible answer. Right, then let's have a look at the different scenario where we're now taking the plastic barrier, we've placed it on the dark side. So yet again, we've got our uniform light, which is coming in from the right-hand side towards the left, and the auxins are diffusing towards the dark side of our plant. However, this is now the Coleoptile's apical bud, and these auxins can no longer diffuse past the mica barrier or past the plastic barrier, and they're kind of like just stuck there. So as a result of this, the plant will not be stimulated significantly on either of these two sides, and the plant will just continue to grow upright. So the amount of cells that we have here is actually not enough to bring about a phototropic movement towards light. So it's just going to keep on elongating upwards. Right, our last one here, they have given us a Coleoptile and the apical buds being removed, and we have this pink section inserted here. This is actually a gel layer. Now, quite often when I ask my learners, what will happen if I put a little gel layer in between it, they will go and tell me, oh, the plant's going to die. Which probably, if it was up to me, the plant would probably end up dying. But
they're tougher than that.
Yeah,
though.
they're tougher than that. So let's just quickly take what will be the most likely answer out of the box and stick to phototropism for this particular question. So we have a gel layer, which is like agar. They can tell you they've used agar as well, which is actually permeable to our auxins, meaning that the auxins can diffuse past that layer. So it's as if that apical bud has never been removed and the auxins will just keep on doing their job. So when we look at the diagram, we can see that these auxins are now able to diffuse through that gel layer, agar layer, and bring about cell elongation on the dark side, which will cause the plant to become positive phototropic and move or grow towards the light source to the right. Okay, here we have another example of a question that we've seen in past papers. We have your uniform light source coming from the one side, and they've given us 3 different plant setups. So this is the normal situation. If I had a plant in my classroom, and I'm sure you've seen this at your own houses as well, if you put a plant close to a window, the plant will eventually grow towards light. So by now you should understand it's because of the auxins. They are like vampires. They move towards the dark side. And they've now accumulated here on the left-hand side, which brings about faster cell elongation as opposed to the light side. And the plant will grow towards the light. So I want you to think about this. If I was a plant standing up right here, and I want to bend that way, what side of my body would need to elongate? It's this side. So in order to grow that way, my left-hand side will need to elongate faster than my right-hand side. So when you sit in your exam, I want you to think, you are a plant and I want to grow towards the light. How would I grow towards the light? And think about it logically. And that will also assist you in being able to answer these questions. So let's have a look at plant number 2 in our little animated GIF. So this guy here has a dark plastic layer that's at the base of the plant. So as we can see, the plant still continues to grow towards the light. So the question is, why is this plant allowed to grow to the light source? Why is it not affected by this dark plastic covering? And remember that the apical bud, which is this part here, it's still intact, producing auxins, and enough of it is exposed for the auxins to be stimulated by the light and diffuse towards the dark side. So cell elongation can still take place and the plant grows towards the light source. But our plant number 3, he's literally in the dark here. He's got a dark plastic covering over it, and it's oblivious to if it's day or night surrounding him. He's got no response to light whatsoever. So pretty much even distribution of your auxins. Remember to actually write that down, that there's an even distribution of the auxins in the apical bud and throughout the tip, which means that there's even growth throughout this plant, and it will just continue to elongate evenly, and it grows upwards in this example. Right, so here we are now on our third concept. So we've done apical dominance, we have done phototropism, and then we get to the one on geotropism. So the word geo should give you an indication as to what this response is from. So we are working with gravity. It's the growth response, well, the growth response towards gravity or away from gravity, depending on if it's the root or the shoot. So it's important to understand that when we have a plant and we put it in the soil and we water the soil, the plant's got no idea where is up. How do I get out of the soil? How do I grow into a plant? So this guy here would have been grown from seeds most likely, and it's dark in the soil. So there's no light that's going to stimulate it to find its way upwards out of the soil. So it has to find a different way of knowing where is the sky, where is the light. And this is where geotropism comes into play. So yet again, all of these 3 examples, it's linked to auxins, and we're going to have a look what happens if we've got a seed, we put it into the ground, we plant it, how is it managing its way through the soil particles. So in the image that we have here, we start off with the definition of geotropism. Remember, the word tropism is just the growth response towards a stimulus. Now we're being specific. This is now the growth response or the growth movement in response to a gravitational stimulus. So we start off by having the shoot section, which is the part that's going to grow out of the soil, and we've got the root section, which is obviously the part that grows deeper into the soil. And then this is the bean itself. So when this bean has germinated, initially what will happen is there will be 2 parts that come out, and the one side would need to grow up, other side would need to grow down. So auxins are sensitive to gravity. So these little pink dots, they represent the auxins within the young Coleoptile or the germinating seed, and gravity will grab hold of them. So as we proceed through the slides, we can see that the auxins are slowly accumulating on the lower side of the plant, which is closer to the center of gravity of Earth. But the response of a high concentration of auxins in the root and a high concentration of auxins in the shoot will have 2 opposite consequences. So here, this is, as we've mentioned in a previous slide, this is actually the shoot section, and it brings about a faster cell elongation on the lower side here as opposed to the top. So if this side elongates faster, it will cause the plant to bend upwards. As opposed to the root section, a high concentration of auxins actually inhibit the cell elongation to a certain extent, while the top will just continue to elongate per normal. So what we find is that this side will then elongate faster and the plant will grow its roots downwards. So there we have it. On the left-hand side, we have the shoot, which is indicating negative geotropic movement because it's growing away from gravity. Here we've got the root. It is showing positive geotropic movement because it's growing towards gravity. So whenever a plant is growing towards or away, we have to refer to it as being negative or positive. So if it's growing away from the stimulus, it's negative geotropism. If it's growing towards the stimulus, it's positive geotropism or positively geotropic. Okay, so this diagram explains the whole process of geotropism. So first off, as we've established, the auxins will accumulate here on the lower side of the plant. So this image actually shows our petunia. It's been toppled over. So if we take our plant, it's now toppled over on its side. So a plant won't continue to just grow along the table. What will eventually happen is this plant will actually start to bend upwards. Now remember that a plant requires light and it wants to be exposed to as much light as possible. So therefore we will eventually see this guy bending upwards. Now, in this image, we can see on the right-hand side that is now the consequence of a plant that has now started to have a growth response. So the top will grow upwards and the bottom will grow into the soil. And it starts off by having that uneven distribution of auxins. Now it will go, accumulate at the bottom, and bring about two reactions. So in the stem section, we've already said that there will be a high concentration on the lower side of the stem, and that will stimulate the elongation of those cells. So if these guys are stimulated, it means this side will grow faster than the top, which will have a lower concentration of auxins, and the plant will bend upwards. Now, basically what we will find then is that it's growing away from gravity. So we call that negative geotropism. Now, the root section that we have here, we find that there's a high concentration of auxins on the lower side of the plant, but this is now going to inhibit that cell elongation. But there's a low concentration at the top of this root section here, which means that cell elongation will still take place. As a result of this uneven growth that we find here, the plant is going to bend downwards, and we said that the plant is now positively geotropic, or positive geotropism has taken place. But always remember to say that it's growing towards or it's growing away from gravity in your answers, and make reference to the concentration between the two sides. So if you just said that there's auxins and it's accumulating at the bottom, you will not be getting your marks. So you always have to state whether there's a high concentration or a low concentration in order to ensure that you actually get those marks in your final exam. Right.
Okay, so let's talk about what else we brought along. This is a Clinostat, which is something that we're not going to plug in because it's going to take forever for you to see what it actually does. But it kind of helps in experiments to remove the stimulus that you are applying. So it rotates really slowly. Yolandi might want to demonstrate very slowly. We thought the sticker was going to be something that you're actually going to see. But it rotates really, really slowly because plants obviously respond to these stimuli and grow very slowly. You're not going to come back tomorrow and see a big difference. And if you think about this like a merry-go-round, those of you who've ever been on one have noticed that as you're going around, you're experiencing gravity in all directions. And that's what the Clinostat, for example, would do. It would actually remove gravity as a factor by allowing the plant to experience it from all sides. Similarly, if you're on a little pedestal and rotating all the way around and light is coming from one side, you're actually going to have all your sides exposed to that light at some or other point. So you don't have unilateral light, you have uniform light with a Clinostat.
I would like to make that reference. You were saying on the merry-go-round, you know those things that you normally find in a park where you climb on and it makes you can run and you have to hold on.
You have to hold on.
Yes, and then when
you feel like you're falling off all the time.
And when you get off of it, you kind of don't know which side is which. You're so confused. Now, this is one of those little, what do you call them? Like
I have no idea.
But you know, when you go to a park, there's usually a thing that you can run around and spin.
Merry-go-round's the
It's
only
like a
thing I can think
merry-go-round,
of.
but like a flat one. So what will happen is the auxins are just as confused. They have no idea where the light is anymore because they are just all over the show within the plant. So they're actually having a great time on the Clinostat.
It's quite trippy for them. So let's quickly understand the Clinostat and the use thereof in experiments, right? So a Clinostat, if you look at the slide there, is going to be usually your control investigation, your control setup, the thing that is rotating slowly, because that rotational movement, whether you have the plant in an upright position like here, where you actually might be applying light and you're not investigating gravity, you're applying light, it's rotating slowly and it's receiving light from all sides. If you topple it to the side, the plant's rotating in this manner, you're basically looking at gravity, because if it's rotating, it's experiencing gravity from all sides. All parts of this plant, the stem and the root, will be exposed to the gravitational pull on all sides all the time. If you then stop the Clinostat and make it stationary, that would be your experiment, because then you're actually applying the stimulus. So here you would see this would be our control setup, because we have a rotating Clinostat. So even though in the setup we have an opening only on this side of the box, which is allowing unilateral light to come in, because this plant is rotating slowly but surely on this Clinostat, it's actually receiving light from all sides, or it's experiencing light as if it was coming from all sides. So you've removed light, you've removed the independent variable, and therefore that is your control setup. Okay? Your control is always the setup where your influence as experimenter has been removed and you expect nothing out of the ordinary to happen. Here we have our experimental setup. We've stopped the Clinostat rotating, it's stationary, and now, seeing as the plant is not spinning around and light is only applied from one side, this plant therefore can respond to that unilateral light. Auxins have enough time to diffuse to one side of the stem, the dark side, accumulate there, stimulate more growth, and the plant responds by growing towards the light stimulus. So what we can deduce from this experiment is that stems are positively phototropic. But then this is a classic one for exams, where you're also asked about little germinating seeds and their coleoptiles, their shoots and their roots coming out, and having a rotating or non-rotating Clinostat. So the same thing applies. If I've pinned these little seeds to a piece of cotton wool and I've put them on a Clinostat that has been tilted, so it's on its side now, a control setup would be the Clinostat that's rotating. So as these little seeds are rotating around, both their little root and their little shoot are experiencing gravity from all sides at all times. Whereas in an experiment setup, that sideways Clinostat will be applying gravity downwards. So the root and the shoot will respond to this because it's coming only from one direction. So what you expect to see after a few days or weeks, you would actually see that in the experiment setup, your roots will grow down. So depending on how you positioned the little seed, so these two were placed horizontally and their roots growing down. These two seeds were placed vertically, so the root just continued to grow down. The stems, on the other hand, will bend upwards because they are negatively geotropic. So here you can see the little stems of the two horizontal seeds growing upwards, and here of the two vertical seeds, the stems have continued to grow upwards. But with our control, what do we expect to see? We actually expect to see everything stay exactly the same, except that they'll be slightly longer. So if you looked at this little seed a day or three later, that length of that root will just be slightly longer, but its growth direction will stay the same because there's no gravitational force to actually respond to. So remember, in an exam, the stationary Clinostat, that is my experiment, because then I'm applying the stimulus. Gravity or light is only experienced from one side. In a rotating Clinostat, that is my control. Think about yourself on a little rondom-tali merry-go-round. I can only remember the Afrikaans word. Okay, so let's quickly look at the other two hormones that we touch on briefly. But firstly, auxins can also be used as weed killers. That's one of their applications that you might not be aware of, but they can be used to kill broadleaf weeds. They don't really affect your smaller or narrow leaf plants like your grasses. So if you have a lawn that you want to keep tidy, you might spray some auxin weed killer on it. Because what does it do? Think again about a high concentration of auxins in stems and roots. If you're spraying a high concentration of auxin on the stem, that's going to create so much growth or stimulate so much growth that it is not able for the plant to sustain it. It's not able to absorb enough water and nutrients to sustain that growth. It's similar to like giving you a ton of bricks and telling you like build something for me in 2 days, and you're probably going to build something, but it's not going to be all that strong because you kind of put quantity into it instead of quality into it. In the root, on the other hand, you know that very high concentrations of auxins in the roots inhibit growth. So even if you sprayed this weed killer on the soil and it was absorbed by the roots of the weeds, they would also die because it actually inhibits their growth. Right. So the last two hormones then quickly. Gibberellins are only involved, as far as you are concerned, with elongating the main stem. So when questions in the exam concern the lengthening of internodes and the main stem growth, you think gibberellins. Woo! So they are responsible for causing flowers to sprout or develop, and they also stimulate seed germination. So here we have a stem. You learned in grade 10 the basic structure of a stem. A node is usually a place where a lateral branch or some other leaf would form, and the internode refers to the area in between. So gibberellins would cause that area to lengthen. Oh, that's yours.
Just to get back to the gibberellins slide, if we can backtrack there quickly.
There we go.
So remember that gibberellins will be for internode lengthening. So yes, the cells will elongate here, but we are working with a completely different area. So when we talk about auxins, that's cell elongation at the top of the plant, but if we are between two internodes, then that will be your gibberellins that will come into play here. So really vital. And I also want to emphasize that gibberellins, G for gibberellins, G for germination. And that's the hormone that's going to be responsible for taking the seed, and then if water is applied, allowing that seed to grow. Now, the reason why I point this out is our next hormone that we have is abscisic acid. And this hormone kind of has like a counter effect to that of gibberellins, and what it actually enforces dormancy in seeds. So these guys here, if you buy a packet of popcorn in the shops, they are not germinating, and abscisic acid will have a role there. So as the concentration of abscisic acid decreases after you've watered your seeds and gibberellins are activated, we find that the plant will then start to grow. So abscisic acid promotes dormancy, while gibberellins actually promote germination. So that's why I make that G connectation there. So ultimately, our abscisic acid, it is a stress hormone. So whenever a plant is in a stressful situation, let's say, for instance, there's a drought, or if it was a case where you've bought a beautiful plant for yourself in the shops, like our little plant here in the studio today, and you kind of forget about it. Eventually, what we will find is that plant's going to start losing its leaves. Now, it's actually quite a sad story behind why the plant's doing it. It's starting to rid part of its body parts, its limbs, in order to retain its energy to its core. And usually what we find is that the plant will have this abscisic acid. It will be released. It will cause the leaves to fall off. And we also see that the plant is putting its last energy into actually making flowers. And then hopefully there's a pollinator that comes past, pollinates the plant, and seeds will form. So abscisic acid is that last chance of survival hormone in order to ensure a future generation. So it's a adapt or die situation. If the plant cannot adapt, it's unfortunately going to die. So during the drought situation, the plant will start off by losing all its leaves because the leaves are metabolically active. And if we rid it from as many leaves as it can, and then hopefully it will rain soon and the plant will be able to push through. Right, so that's one of the consequences or one of the functions of abscisic acid. Now, there could be other conditions like freezing cold, high salinity water, maybe you're using borehole water and you live close to the beach. The same thing will apply here. The plant's going to have a high concentration of abscisic acid. But we also see a case where you've got fruit hanging on a tree, and we can't have those fruit being on the tree forever. Eventually they will actually start to rot on the tree itself. So abscisic acid will cause this fruit to actually drop off, and then hopefully it falls in a space of soil where it will then germinate and a new plant will start to develop. So abscisic acid will bring about abscission, which is when the tree actually rids itself from its leaves or its fruit. Right, so let's go through our slide quickly. So we started off with the stressful environmental conditions, the functions, as we've said. So it's a growth inhibitor, so it will not bring about cell elongation. The plant will actually be stunted in growth. We've got abscisic acid causing the stomata to close. So in this case here, we have a drought situation. All the stomata and the leaf will actually close up, or most of them, to try and conserve as much water as possible. Then we have dormancy being promoted in our seeds and buds. So here, for instance, that little lateral branch that we have there, that little bud, that will be inhibited from growing as well. And then, as we said, allows for abscission. So during the autumn, when the leaves start to fall off, that abscission where the leaf drops off, that is as a result of the hormone abscisic acid. Right, then we're just quickly going to finish off with the last couple of items on our to-do list, so to say. And this is chemical defenses. So within a plant, there are two main defense systems that they can take. One could be a chemical defense mechanism, and the other one could be a mechanical defense mechanism. So in the image that we have here in front of us, we can see that there's a warning sign attached to this tree, where this particular tree's fruit, it actually contains high amounts of cyanide in it. So if a person had to eat this particular fruit, maybe they've ended up on this beach, they could actually die as a result of it. And it's not just cyanide, it can be strychnine that is in the plant itself as well. Quite often, the plant could have a bitter tasting chemical inside it, like your oils, mint, for instance. That's also like a bitter taste to some animals.
I mean, we as humans, we use a lot of these essential oils. Those of you who know, lavender and tea tree, they all have these, medicinal properties. Tea tree is antifungal and antibacterial, so it's part of your face wash. And plants are also, they have to defend themselves against, bacteria, viruses. They have to defend themselves against herbivores. I mean, that's their form of a predator,
And some animals and insects, they are adapted to be able to eat these plants with these essential oils in it. While you have others that they taste it and they're like, no, this is not for me, and they run off. So that's the plant's attempt at preserving its life by secreting a substance that makes it unpalatable for the organism that's eating it.
And preventing overgrazing.
Yeah, overgrazing. And
something very interesting with the acacia tree
I was
is...
just about
yes,
to
are you going to?
mention
Okay,
it.
fantastic.
No,
You
go
do
for
it.
it.
Okay. The acacia tree, when giraffes actually eat from it, then they start producing... Do you know what the chemical they produce?
It's tannins.
Tannins. And it basically happens in a few minutes,
Yes.
and they graze for a few minutes on the tree, and then it starts tasting unpleasant for them. And then they start moving on to the next one. But what's very interesting is that acacia tree that has now been eaten from actually doesn't just produce tannins in its leaves, it also releases a chemical into the atmosphere.
Yes.
That's then sensed by
Other
neighboring
trees.
acacia trees in a vicinity. So even before the giraffe has started eating from that tree close by, it's already started to taste bitter. So that actually forces the giraffe to graze farther and therefore not focus too much on one area of a particular forest.
So usually they will either move on or they can go upwind where the chemicals haven't reached. So they're actually quite clever. They know where to go. Or they can just skip 7 trees or so and then start grazing on another, browsing on another tree.
But it's a bit freaky to think that you move into the Amazon forest and like the plants are aware of your presence far before you've actually gotten to them.
They're communicating with each other. It's quite
that's chemical defenses, eh?
Yeah, and also one of the consequences of some of these chemicals is that they actually inhibit digestion. So the tree's kind of like having its last laugh by ha ha ha, you've eaten me, but I'm not going to let you gain any nutrients from the proteins, the plant proteins inside it. So yes, definitely like a protein inhibitor or digestive inhibitor as well, which is like, I think it's amazing, like how these plants work.
Very intelligent creatures.
Yes.
I prefer them to humans sometimes.
Right, then these are our mechanical defenses, and I'm sure that you guys have all experienced this somewhere along the line. I used to have this little succulent on my table with these really big thorns on it, and you would have people that come up to it and like, oh, this is pretty, pick it up, look at it, and they end up pricking themselves with one of these thorns. Now, that's actually the plant's like big aim. It sounds a bit sadistic, but that's his way of ensuring its survival. So as soon as you've pricked your finger, you've exposed your skin, which means that the pathogens have an entry point into your system and could potentially end up being an infection that then...
And there are also some examples of symbiotic relationships between these thorn-like structures and, bacteria or fungi that live on it. So the plant has this symbiotic relationship where it's kind of protecting itself and the bacteria get some host to grow in.
Yes, definitely.
And you get some people who are prickly as well. So, yeah. That's about what we have time for today, Yolandi.
That's right.
So hopefully you understand plant hormones a little bit better. Being teenagers yourselves, you can familiarize yourself with hormones. They have other functions in plants, but they're still there. And make sure that you go and learn these little boxes, these silos, as Yolandi says. Auxins have this and this and this function. So when I get a question about light, I'm going to focus on phototropism.
And then also in our PowerPoint presentation, which will be uploaded onto the website, we've included a few questions for you guys to work through with the memo included. So see if you can apply yourselves and whether you find those questions challenging or not.
And that is then our last grade 12 lesson for the year.
Right.
So challenging times ahead for you. Apply yourself, study well, and good luck with your final exams.